So throughout all our discussion, we're not going to make much reference to transport. But remember, all this movement that we're talking about requires energy. It requires typically the motor proteins of the cytoskeleton. And we talked about this for virus particles getting into the cell, whether it's a naked virus or an endosome moving from the plasma membrane towards the nucleus, that requires movement along microtubules. And it's the same for components for assembly. They have to go towards the nucleus or away from the nucleus on these tracks. So this is long-range transport that requires energy. Now this movement, besides all the characteristics that I've told you about, this movement is facilitated by addresses built into viral proteins. So for example, you know, of course, all about signal sequences that direct membrane proteins to the membranes through the ER and the Golgi to the plasma membrane. There are also fatty acid modifications that we'll talk about later that help send proteins to the membrane. There are retention signals that keep proteins in certain places. If you want a protein to stay in the ER, you have an ER retention signal on the protein. So some viruses actually bud from the ER, so they would have these signals. Nuclear localization signals for proteins that need to get in the nucleus. Nuclear export signals if you need to get out. And finally, again, we'll come back to microtubules and filaments as ways of moving proteins around. So these are clearly involving viral protein interactions with the motor system. And much of that remains to be sorted out. So here's just an example of nuclear localization. And again, it's something that most of you are likely familiar with. We have a cell infected with a virus, needs to replicate in the nucleus. And there are three examples shown here: influenza virus, polyomavirus, and an adenovirus. So influenza virus, you remember, replicates its genome in the nucleus, and it starts to assemble the ribonucleoprotein there. So the nucleoprotein, which is a component of that RNP, is made in the cytoplasm on free ribosomes, and it gets shipped into the nucleus. The nucleoprotein has a nuclear localization signal, a very short amino acid sequence that specifies to the transport apparatus, I need to go in there, and that's how it's taken up. The same for other viral proteins that need to get in the nucleus. Here we have some structural proteins of polyoma or adenovirus needing to get in the nucleus because their DNA, the DNA of these viruses is replicated there, and the virus is going to be assembled in the nucleus. Why is not clear, but I suppose it makes sense to assemble the particles where you've replicated the DNA genome. So these capsid proteins also have nuclear localization signals that specify them for import. So that's an example of getting the viral structural proteins to the right place in the cell using these kinds of addresses. In the same way, many viral glycoproteins, remember envelope viruses have glycoproteins in their membranes. Those are typical proteins made in the secretory pathway. So they're made on the rough ER, shown here, the ribosomes all bound to the outside of the ER. The protein is translated through into the lumen of the ER, typical membrane glycoproteins. And then the glycoprotein needs to go to the surface where it will eventually participate in budding of the virion. And to get there, it goes through the secretory pathway. It moves from the ER through the various stacks of the Golgi by vesicular transport, and then eventually up to the surface in a vesicle, in a secretory vesicle. And that vesicle fuses with the plasma membrane, and now we have a viral glycoprotein at the cell surface. And this is going to serve as a nucleation point for other structural proteins that need to be incorporated into these particles. As you can see here, other viral proteins are joining it. And look, the movement of these secretory vesicles, again, requires microtubules, transport from one place to the other. So here's an example of the need for the microtubules in the movement of a viral protein. This is a cell infected with vesicular stomatitis virus, that bullet-shaped virus related to rabies virus. And remember, its RNA genome is packaged as a helical nucleocapsid. The RNA is bound to nucleoprotein, the NP. There's one copy of this protein repeated many times across the length of the RNA. And this is an infected cell, as I said. The nucleus is stained red. The microtubules are stained, sorry, that's blue, not red. The nucleus is stained blue. The microtubules are stained red. And the viral nucleoprotein is stained green. So you can see in the top panel, this is just an infected cell, untreated. These nucleoproteins are moving from the periphery, from around the nucleus towards the plasma membrane. So they're moving there by transport on microtubules. They're going to make it to the plasma membrane and eventually be incorporated into new virions. If you treat these cells with nakitazole, it's a drug that disrupts microtubules. That's shown in the bottom. You see now the nucleoprotein is all accumulating in patches. It's not going anywhere. So wherever it's made, it's just accumulating and aggregating. So this shows you one bit of evidence that you need these microtubules to bring the nucleoprotein to where it needs to be, which in this case is the plasma membrane.